GeneMarker HTS software provides rapid analysis of STRs, Y-STRs, and mitochondrial DNA data from massively parallel sequencing systems like the Illumina and Ion Torrent platforms for forensic applications such as database or casework samples, all of this in an easy-to-use Windows operating system with password-protected user management and audit trail. The STR application allows for analyzing autosomal and Y-STRs using forensic nomenclature. Allele calls are concordant with CE results, but also include sequence variants to visualize iso-alleles. The mitochondrial DNA application allows for analyzing whole mitogenomes or specified regions with multiple alignment options as well as forensic nomenclature and the ability to upload profiles to MPOP. Starting a new analysis is easy. Enter a location to save your reports, use the default reference and motifs, or import your own. If you are using one of Promega's PowerSeq kits, you can choose to use the corresponding panel, otherwise select None. Today we'll be looking at STRs, Y-STRs, and mito data that was generated using the PowerSeq 56 GMY kit. To the right are alignment options for mitochondrial DNA data analysis. And down at the bottom are filter settings for both STR and mito analysis. Once all of the settings have been made, click OK. As samples finish, they will become available in the viewer window. You can open up and view a sample while the others are being aligned. This is the STR results screen. On the left is the histogram viewer, showing separate histograms for each of the STR and Y-STR markers. The different colored bars indicate the presence of iso-alleles. Iso-alleles are loci that appear homozygous in length-based measurements like capillary electrophoresis, but are actually heterozygous when sequenced. On the right is the STR results table that includes columns for the allele name, filter information, fragment sequence in a bracket format, and forward and reverse read counts. The read depth information can also be imported into master probabilistic genotyping software for mixture analysis. The table and the histograms can be exported separately for your records. Mitochondrial DNA results are available by clicking on the Mitochondrial Alignment tab. At the top is the global viewer, which displays the depth of coverage with forward reads in blue and reverse reads in red. Below that is the reference sequence, in this case the revised Cambridge reference sequence, and the consensus sequence. In the center are green blocks indicating the location of the motif regions and blue blocks indicating amplicon regions. In the center of the screen is the pileup of reads, including arrows indicating the forward and reverse reads. To zoom in, draw a box from left to right, scroll by holding the right mouse key and dragging, zoom out by drawing a box from right to left. To sync the global viewer and the pileup, select Zoom Global. The green vertical bar in the global viewer indicates a difference between the reference and the sample. In this case, it's showing an A to G SNP. The very bottom is the variant table, which gives the user the option to view variants for the major profile, minor profile, or both. Results are saved automatically when the sample is initially analyzed. Any edits to existing projects must be saved as a new report. Doing so will also save the edits as part of the audit trail in GeneMarker HTS software. Result tables for each sample include BAM files, consensus sequence, major variant, minor variant, and alignment statistics log. The major report has the format needed to copy and paste the variant list into the MPOP database for haplogroup information. Thank you for your interest in GeneMarker HTS software. For more information or for a free 35-day trial of GeneMarker HTS and other SoftGenetics products, please visit www.softgenetics.com or email info@softgenetics.com. For technical support questions, please write to tech_support@softgenetics.com. Thank you for watching. The green vertical bar in the zoom global field indicates a difference between the reference and the sample. In this case, it's indicating an A to G SNP.